Good morning, everybody. This is Zach Litvak from Swedish Neuroscience Institute here in Seattle, Washington, welcoming everybody to Neurosurgery Grand Rounds. It is my absolute pleasure this morning that we are joined by Dr. Analiz Rodriguez from the University of Arkansas. Dr. Rodriguez is a MD PhD. She completed her medical science training program at Case Western, and then proceeded to neurosurgery residency at Wake Forest, then completed her neurosurgical oncology fellowship at City of Hope Hospital, where her research focused on glioblastoma and immunotherapy, and then joined the faculty at University of Arkansas, where she has risen very quickly to associate professor. Dr. Rodriguez has a number of research interests, but will be speaking to us this morning on the potential game changer impact of long-read sequencing applications for brain tumors. Dr. Rodriguez, thank you for joining us. And as we were discussing before, glad to hear that things have calmed down weather-wise in Arkansas.
Well, thank you so much. I really appreciate the invitation. I'm going to go ahead and share my screen. Can you see that in presentation view?
We do. You're good to go.
Perfect. I'll go ahead and get started. So today I'm going to be talking about long-read sequencing applications for brain tumors. And do not worry, it is going to be a lot of genomics, but I'm going to talk about it from a perspective of a neurosurgeon. So that means it can't be too complicated. No, but more complicated than for orthopedic surgeons. No, I'm joking. So I have a couple of disclosures. These are my disclosures. And then hopefully by the end of the talk, you will be able to understand the difference between next generation sequencing, which is the most common form you're probably familiar with, and then this new form of sequencing, which we'll talk about today, and then think about some of the clinical applications and what will happen in the future when I believe this will become clinically integrated. So like I mentioned, I'm going to just review a brief genomics timeline, but from the perspective of a neurosurgeon. In 2003, that was actually when the Human Genome Project ended. This, set out to sequence the human genome. This project started in 1990, and then it was completed in a little bit over 10 years. So in 2003, we had the human genome sequence. Also, in 2005, the Stoop protocol was released, and that obviously is very relevant for us. That was the year that we started to manage patients with glioblastoma, with temozolomide, and radiation. Interestingly, that same year is when next generation sequencing came out, and that next generation sequencing, also known as Illumina sequencing, also known as second generation sequencing, is still used today. And that basically allows you to clinically sample a lot of different DNA fragments in tandem. And that is a very powerful tool. And then in 2008, the Cancer Genome Atlas came out. And a lot of people don't know this, but glioblastoma was the first cancer in the TCGA. So I think it just kind of shows how much brain tumors have really been relevant and at the forefront of genomics and cancer genomics specifically. So that to me is always very interesting. It's not lung cancer, it's not breast cancer. GBM was the first cancer in the TCGA. And then in 2016, the World Health Organization released an update on brain tumor classifications. And for many of us, we're familiar that now there's also the 2021 WHO guidelines. But what stands out about the 2016 guidelines is for the first time, they started to require molecular markers. So before 2016, diagnosis was based on histopathology findings. And then in 2016, our world changes, and now we're starting to need to understand what genetic mutations are present in the tumor. And this actually really is a game changer, because histopathology can be done anywhere. Anywhere in the world, you just, have a slide, you look at the characteristics under a light microscope. But now you're requiring genetic sequencing, and that may not be available at a lot of places. And you may not be able to do that at your own hospital. You have to send a sample to someplace that has sequencing technology and capability. So I go to this is a famous paper that came out in 2012 by Michael Taylor. Many of us may remember this came out when I was a resident. I don't want to age myself too much, but yes, in 2012, I was still a resident. And I remember this coming out because before this, I was memorizing all this histopathology and remembering Homer Wright rosettes for medulloblastoma. And then this paper comes out and we learn that there are 4 molecular subgroups of medulloblastoma, with W and T having a really good prognosis and group 3 having a poor prognosis. And you can see that these different subgroups have various genetic drivers. This impacts therapy, this impacts prognosis, this impacts what groups of patients have this. I really like this from the paper that they talk about, well, at this, and remember, this is in 2012. So they say, well, histology and WHO classification has not been supplanted. But it's clear that histology alone is inadequate for the diagnosis and classification of medulloblastoma. So this is back in 2012, before those 2016 guidelines, but you already have kind of know that this is coming. This no longer are we just going to think that histology is it alone. And just as a reminder, this is the WHO classification from 2021 for gliomas. Now, this is very much super complex now. Things that stand out to me of significance is that glioblastoma is now only a term applicable to IDH wild type. And before, grade 4 astrocytomas also we thought included IDH mutants, but now IDH mutants and IDH wild type are totally different. Histologically, I can't tell, IDH mutants or IDH wild types apart. A machine could, because there is a paper out about machine learning and artificial intelligence and using histopathology, feeding algorithms, and a machine probably could, but a human probably cannot. And then the other thing is our understanding of, for example, diffuse midline glioma and these histone mutations and histone variants and how that impacts pediatric brain tumors. So lots of changes coming in that came in 2021. And then this is just another example. Here is ependymomas. And so ependymomas can occur in both the pediatric and the adult population. They can occur in the supratutorial, the infratutorium, and the spine. And again, now we have all these subclassifications, and you can see that these oncogenic drivers and genetics are really helping us differentiate these tumors. And then again, it's really important for us to understand this, because when you're counseling patients and you're talking to them about, well, what is your prognosis? Now you really need these molecular markers to guide that, and that guides your management as well. Another thing that's emerging is liquid biopsy. We hear about this a lot. Liquid biopsy is now integrated in a lot of oncological paradigms, most commonly in lung cancer. So now in lung cancer, that has been, FDA approved, and you can get liquid biopsies throughout your treatment to track how you're responding to therapy, and potentially even in some cases to diagnose you with certain mutations that are present in your tumor. Liquid biopsies for brain tumors is not yet in clinical use, but definitely a lot of clinical trials and preclinical information about this. So what is a liquid biopsy? Basically, it's analyzing cell-free DNA. So tumor cells, they die, they release DNA fragments. And these DNA fragments are released both into the spinal fluid as well as into the blood. Of course, for brain tumors, there is the blood-brain barrier and the blood-tumor barrier. So the cell-free DNA is probably not as high a concentration in the blood as other tumors, but it still can be detectable in the blood. And then in the spinal fluid as well. And that, I don't think that's surprising to us at all, especially when you deal with leptomeningeal disease. Not surprising that we can assay the spinal fluid and get information. Now, I was talking a lot about molecular markers, and that is just identifying DNA mutations. But obviously, things are a little bit more complex than just reading a DNA strand and identifying a mutation that's present. There's also epigenetics. And epigenetics just is a broad term in terms of different changes that can occur that affect both transcription and translation. The most common thing that you can think of is DNA modification, specifically methylation. So we're very familiar with methylation of cytosine, and that usually occurs at CpG sites. So you have a cytosine and guanine dinucleotides. These are typically at the promoter region of a gene. And when you have a lot of cytosine methylation at at these CpG sites at the promoter region, this can indicate to various transcription proteins whether or not to skip this part of the DNA or to go ahead and transcribe it. So basically, it's a way to turn on and turn off genes. And that's very important for obviously development, but then also in cancer. The other thing is chromatin modifications. Obviously, your DNA is super, super long, but it's wound up along histone proteins. And these histones have different modifications, and they decide to wind your DNA up into tight balls or elongate your DNA, allowing for transcription to occur, allowing for proteins to come in and start reading the DNA. But they, depending on their modifications, they can decide, you know what, I'm going to wind up this DNA very tight. You're not going to get in, you're not going to read these areas. And that's another way you can turn off genes. And then thinking about downstream of DNA and RNA, we have a lot of non-coding RNA, silencing RNA, long non-coding RNA. And then interestingly enough, we have RNA modifications. So you can have methylation, for example, on RNA. And now that's a new field opening up, epitranscriptomics. So a lot of things going on. How does that affect us clinically? So meningiomas, this paper came out not too long ago, that if you were to look at methylation groups of meningiomas, you can actually classify meningiomas into various groups. And there are 3 main groups, and this really ties to recurrence and prognosis, and is not necessarily correlated with histopathological grade 1, 2, and 3 that we're familiar with. And so now we're starting to think of meningiomas very differently than before. And this may impact down the line a lot of these trials. For example, current guidelines for meningioma. If you have a grade 2 meningioma and you had a gross total resection of a grade 2 meningioma, the guidelines say that, you know, you can go either way. You can advise a patient that, yeah, I do think you need adjuvant radiation, versus you can tell a patient, you know, I got a gross total resection. It was a grade 2, but I'm just going to follow this closely with MRI. I think in the future, we may get a methylome and tell a patient, you know, you're at high risk for recurrence. I had a gross total resection, but I'm going to give you adjuvant radiation because of this molecular markers. So again, this is definitely a game changer. Now, I mentioned to you how methylation is basically a very powerful way of a cell turning on and off genes. And so you can get a pattern of what genes are turned on and off. And this is very specific. This pattern is very specific to what type of tumor is present. And you can see this is a cluster diagram where tumors that have more similar patterns are clustered closer together. And you can actually see that glioblastomas versus ependymomas versus pineal tumors are clustering in different areas. And this is by looking at their differential methylation regions, so DMRs. And this is actually sometimes a little bit easier to figure out than, say, looking at one gene that may be at a low frequency. And sometimes that's hard to find if you have a low amount of DNA. But if you're able to look at someone's pattern of what's turned on and off across, you can read all different parts of the DNA and just look at the pattern. And then maybe from that alone, you're able to figure out what type of tumor it is. Interestingly, this is actually being used in very difficult cases. So neuropathologists now, sometimes they're not able to give a diagnosis based off their current tools. And sometimes you can go ahead and send off a methylome, and that can help you differentiate what type of tumor it is. It's not yet clinically, integrated to the point that we're doing this on every case, but our neuropathologist here definitely does send out the methylome when it's just, I can't tell, this is a complex glial neural tumor, and I actually can't really tell what this is. The thing about looking at methylation, though, is that it takes a lot of time. And this is actually from this paper from a German group that is really great. And basically, you can see that it takes at least a week. And the DNA, you can't, with next generation sequencing, which is our most common form of sequencing, you can't just read the DNA strand and figure out where the methylation groups are. You actually have to treat the DNA with bisulfide, and then this will change the cytosine to a thymine, and then you can do PCR, and then you can figure out where these methylation groups are. And we'll talk about how long read sequencing is actually better than current methods, and we'll get into that a little bit later in that talk. But this is an example of a methylation profiling report that you can get, and you can actually submit, research samples or other clinical samples if you do end up getting a methylome, and look at put it into this database. This is molecularneuropathology.org, and then you can kind of see where your patient's tumor clusters and kind of predict maybe what type of tumor this is. Of course, there's a word of caution on this website. This classification using methylation profiling is a tool for research only. I do think, though, that's going to change. I think that's probably in 5 to 10 years. I predict that once methylation profiling gets a little bit cheaper and easier to do, I think this is going to change, and this will no longer just be for research. It'll probably be integrated clinically. So one of the most common epigenetic markers we're used to talking about is MGMT methylation, which MGMT is a DNA repair enzyme. And then we know that when MGMT promoter is unmethylated, then you get DNA repair, and temozolomide doesn't work as well. However, when you have a methylated MGMT, that silences the MGMT DNA repair enzyme, allowing TMZ to do its DNA damage and allowing TMZ to work. So we know that when we sequence these patients, we get MGMT promoter information, and we tell people, oh, you're methylated, you're more likely to respond to temozolomide. And then the histone, I talked a little bit about histones. This is actually clinically used now for understanding midline gliomas. So histone 3 mutations are now integrated into identifying different types of midline gliomas, mainly in pediatrics and young adults. And then no surprise, we were talking about gene silencing, and this is very much involved in neurodevelopment and how cancer progresses. And this really, we're starting to understand the cells of origins of these really complex tumors. And this is all, new and developing, understanding where these cells are arising from, what is going on in these young children's brains, that these developmental programs got thrown off and ended up forming a tumor. So I talked a lot about the methylome in tumor tissue, but what about if we looked at cell-free DNA in a liquid biopsy? And what's really interesting is a lot of times I kind of told you, yeah, well, because of the blood-brain barrier, the brain tumor DNA is not going to be shedding into the blood at the same rate as other type of brain tumors. And so you have a low quantity, and that's going to be really hard to identify a DNA mutation, especially if it's a low frequency. But what if you looked at the methylation pattern? If you were able to just sequence whatever DNA you have available and then looked at the pattern of methylation, does that cluster similar to when you do it on tissue? And the answer is yes. So this is work from Dr. Zade's lab in Toronto, where her lab is showing, yeah, it does seem that here we are taking blood samples from brain tumor patients, isolating DNA and just looking at the methylation. And we're able to see that these different types of tumors are clustering. And can we use that diagnostically? And the answer is, yeah, we can. And again, what's so powerful about this is from blood. This is not spinal fluid. So again, not very invasive. And there they are showing how they're able to differentiate a hemangioparasitoma from a meningioma, a low-grade tumor, IDH mutant from IDH wild type. So potentially in the future, could we be offering liquid biopsy over brain biopsy? And that is potentially possible in maybe 10 to 20 years, and that's very exciting. So a little bit about the sequencing evolution. I kind of gave an overview about how molecular markers began being integrated officially in 2016. We've seen a huge evolution of this and so rapidly. And the same goes for the sequencing technology. So first generation sequencing, also known as Sanger sequencing. Many of us did this in the lab when we were in biochemistry in undergrad. And then Illumina sequencing, second generation sequencing, still very much used. And here we're putting DNA fragments into a flow cell that has these oligos, and then these the DNA fragments kind of line up on these flow cells, and then you do bridge amplification, and then you do sequencing by synthesis, where these tagged nucleotides, you're getting these fluorescent signals, and you're reading, and you can read. It has a high accuracy, and you can read a lot of fragments at the same time. And so this is really pretty much the most common form that's used clinically, is this Illumina sequencing. Third generation sequencing, also known as long read sequencing, also known as nanopore sequencing. I know there's a lot of terms, but I'm just, for the purposes of this talk, going to call it long read sequencing. What's really cool about this is you take the DNA and you pass it through a pore that fits the DNA molecule. This pore is embedded into an electroresistant membrane. And as the DNA travels through, it changes the electricity of this membrane. You actually get a readout, and you get this little graph, and this raw signal can be deconvoluted, and you can do base calling. So you can say, oh, this is an A, this is a C, etc. What's even cooler is it can read a DNA strand that you haven't altered in any way. You haven't done any chemical thing to it. You haven't amplified it. You can just read a naked piece of DNA. You don't have to break it up into pieces. It can read up to 2 million nucleotides. So you can read long fragments, and hence its name. But what else is really cool is the signal for a cytosine is different than a methylated cytosine. So then you can get methylation straight from the DNA without doing any bisulfide treatment. And that's very powerful. And so this is an example of the pore embedded into the membrane. You can see DNA passing through. You can also pass through RNA. And most recently, there's some of these pores that you can change the size of them. You can even theoretically, and some people are doing this, put proteins through this and read peptides by peptide of a protein. So that's really awesome. What's also is really cool is these are portable. So this is actually what one of these sequencing devices looks like. You can just drop in this. This is actually the real size pretty much. You can just drop in your DNA and then you are sequencing in this tiny device, and it has a USB port. And then you just put it into your computer and you get those electrical signals, and you can deconvolute on your computer. So this means that you can take this little portable sequencing device into, for example, if you're in, the Amazon rainforest, you theoretically could sequence something as long as you have a computer. So, all right, so we're going to go through some clinical cases to highlight where the utility of long-read sequencing can come into play for brain tumors. So this is a patient who came in with seizures. They actually had newly diagnosed renal cell carcinoma, and then these multiple enhancing brain lesions that you can see here. Ended up not being sure if this was metastasis or if this was GBM, and took this patient for a biopsy. Obviously, some of these lesions are in the motor strip, so didn't want to just jump to, surgical resection. Biopsied, it was very bloody, so at a certain point had to stop, and ultimately the pathology, histopathology, came back as mild glial proliferation with atypical cells. Unfortunately, that's not really diagnostic. And then we had to wait 2 weeks, and we got next generation sequencing results. And this patient had a TERT promoter mutation, and therefore this got upgraded from, glial proliferation to a GBM. The problem is, though, this took about 2 weeks to get that information back. And so we'll talk about long read sequencing, how we can really decrease that time. Here's another example. So this patient is a 37-year-old patient who had an IDH mutant grade 4 tumor resected. She really is functionally doing well, still ambulatory, just some mild right-sided weakness. This huge tumor, she doesn't qualify for any clinical trials. A lot of the recurrent high-grade glioma trials have a size cutoff. And she was trying to find a neurosurgeon that would take her to surgery. I ended up taking her to surgery and also implanting gamma tiles so that she had brachytherapy. But something that I usually do is that I actually do molecular characterization at each stage. So I requested her pathology from the other hospital where she had her first surgery. And this is an example of the clinical next generation sequencing report. And then this is a surgery that I did. And I use this to show that this is no longer the same tumor. So you can see that her tumor mutational burden, which is an estimate of the mutations per megabase, was initially 4.2. And then she comes back, and this is a hypermutated tumor now. Her mutational burden is now 62. And that's in the 99th percentile. And you can just see that all of a sudden she has all these new mutations that weren't present in the first tumor. And so I really think that it's important to understand that our brain tumors are influenced by treatment and influenced by just their natural evolution as they progress. So it probably would be worthwhile to continue to do molecular characterization at each time that you do perform a surgery, and that, following tumors over time will help us understand how does resistance to treatments actually occur. And then this is another case. So this patient came in, a 24-year-old, with a remote history of colon cancer at a young age. He came in with headaches with this large tumor on the left side, resected. Everything went well. Thank God he didn't have a post-op speech deficit. He underwent chemo and radiation. And then this is him 3 months later. Not only does he have local recurrence, he has distal recurrence on the other side. Our radiologist, noted this enhancement on the other side along the dura. I ended up taking out both lesions. You can see, yeah, that little tiny dural base lesion is a distal recurrence. So what's interesting about this case is histopathologically, this ended up being, a glioblastoma, but our pathologist noted, it's not a normal glioblastoma. I see features of ependymoma and pleomorphic xanthoastrocytoma. And so you can see histologically, there's these different regions of the tumor that make you say, wow, this is atypical to just a classic run-of-the-mill GBM. And so here we did comparative transcriptomics, which is looking at the RNA profile and basically the pattern of gene expression, which I kind of harped upon earlier about the methylome gives you a similar output as well when you look at the differential methylated regions. And similar to those maps that we looked at earlier, if a tumor, each dot is a tumor, and if a tumor is similar to another tumor, it's going to group together and be close in a cluster. And so this tumor was really strange in terms of its clustering with ependymoma, with low-grade gliomas, and with GBM, but no tumor had a greater than 95% correlation. And our patient had a Li-Fraumeni syndrome, which is a germline mutation in P53. So very unique tumor. He ended up being super young. We were very aggressive. You can see he has a myriad of treatments. And this, I just highlight one of his surgeries. You can see he has this periventricular enhancement. This ended up coming back as fibroid necrosis without tumor. Something is very common for us neurosurgeons that, we take someone to surgery and it's not a recurrent tumor. And it's really hard to differentiate. We use MRI, we use spectroscopy, perfusion, but ultimately the only way you're going to know is right now is a tissue diagnosis. But that is invasive, whether or not you're doing a biopsy or resection, that can be very invasive for the patient. So again, methods to monitor treatment course that are better than MRI, we really need that. And so we're going to go ahead and talk about how long read sequencing can address some of these issues. So first, what about that real-time diagnosis? I don't like to wait 2 weeks to figure out what type of tumor I'm treating. It also really delays enrollment into clinical trials. And sometimes if you're part of a multi-institutional clinical trial, you do the molecular characterization and then you have to send the pathology block to the central location and they redo their own sequencing and it can take a long time. So in this is work from my lab. Well, what we ended up doing is using Cas9 to just take DNA directly from a tumor sample. We are not amplifying the DNA. We are not doing any chemical treatment. This is just raw, naked DNA, as I call it. And what we're doing is that we're cutting out the regions we're interested in. In this case, it was MGMT because that's a prognostic biomarker, and then IDH mutations. We were able to do that in multiple clinical samples. And this was very much in line with results that we get with other forms of sequencing, Illumina and Sanger. And then we were also able to look at this MGMT promoter region, but in a much, much better way than what is clinically used. Currently, there is no standard way to assay MGMT, meaning that some various MGMT testing sites. They use various methods, but not only that, some people are not necessarily looking at the same CPG regions in the promoter, looking at the same number, and then also the level of methylation, the percentage cutoff may be different between tests. So one test may say if you have over 50% of these CPG regions methylated, that's a positive methylation. Someone may have a lower cutoff. That's an example. So here you can see with this is cell lines as well as patient samples. We're able to look across the promoter region, including the intron and the exon, and get these beautiful methylation data. So again, ideally for this type of advanced long-read sequencing, you do need fresh tissue, but you could theoretically do DNA, RNA, and get epigenetic information. And potentially, you can improve your protocol to get results in the same day. And that's something we said at the conclusion of this paper. And then I saw another paper that came out not soon after. This is a group from Oslo, and they really are doing this the same day. What is awesome about this paper is that they're doing it and giving you intraoperative diagnosis. So they were able to, their paradigm is really awesome. So they work with the neurosurgeon. As soon as you get to the tumor, like normal, you're going to send a frozen specimen, a little chunk of the tumor. You're sending it now to these sequencing people as well. And they're prepping a library and they're giving you a report back within a couple of hours, sometimes, within 3 hours, and you may still be resecting that tumor, and that could potentially influence your paradigm. So here is an example of things that they were able to use this for intraoperative diagnosis. Again, I love this paper. It's so cool to me. Not only, so for example, they talk about this case where they're able to tell the surgeons that this is a medulloblastoma WNT versus a medulloblastoma sonic hedgehog. What they found is that a lot of times this intraoperative frozen sections were inconclusive, and sometimes and that their intraoperative long-read sequencing really matched the final pathology almost all the time, and that sometimes what they were able to tell the surgeon actually impacted the surgical strategy, because now as a surgeon, if you know what type of tumor it is, you can decide, do I need to go for broke and go for a gross total and risk a neurological deficit because that is the best chance my patient has? Or, you know what, I can hold back. I know what type of tumor this is. It's actually really responsive to chemo and radiation. I'm not going to go for broke. So very powerful technology. Here's another example using long-read sequencing for liquid biopsy. This is from CSF. Again, this is in clinical samples. This is a pediatric glioma cohort. And what they were doing is they're serially sampling these children that had pediatric glioma, and they were on a special trial getting a new agent, UNC201, that's a dopamine, targets a dopamine receptor. So these are children with histone 3 mutated diffuse midline glioma. We're very familiar that this has a poor prognosis, very difficult to treat given the location of the tumor. So they're oftentimes not able to do a gross total resection. And here they're following these patients over time. Patient B, this patient actually got this agent and did not have on radiographic clinical response. You can see the MRIs over time. That tumor volume is not really changing. That matches basically, if you look at the graph below, this cell-free DNA. You can still see that cell-free DNA levels pretty much stay the same when you're looking for that marker of the glioma. However, in contrast, this 17-year-old female, she actually did over time have regression of the tumor up to 40% of the volume decrease on this UNC201 treatment. And then what's really cool is that the CSF profiles of cell-free DNA. You can see the concentrations of the cell-free DNA go down. And so is this a way to more minimally invasively, rather than sampling the tumor, kind of understand what's going on in the brain? So this is another example. This is long read sequencing for ALL. ALL can actually relapse in the brain, and that's really bad for these children. There's a lot of limited options for ALL once it relapses into the brain. And so in this study, they were looking at blood as well as brain CSF samples. And what's really cool about this is they were able to show that you can sequence, using long read sequencing, you can identify cell-free DNA in as low of quantity as 25 picograms, which is really amazing and shows the power of this long read sequencing. These are patients. They identify relapse in the brain, similar to how we identify leptomeningeal spread. This is either by cytospin, a cytology, or flow cytometry. And so they looked at patients A and B. So in the top part, these are patients who were known to have CNS relapse, as defined by positive, blast cells in the CSF by flow cytometry. And so they sequenced the bone marrow, so that's the gDNA. And then you can see that at the time of diagnosis, when they did flow cytometry, you can see that they were able to identify cell-free DNA in the CSF as well. And they can actually look, this is the immunoglobulin gene, they can actually look at the different clones that are present. Clonality in ALL is really important because that tells you what your treatments can affect certain clones of the tumor. And so it's really cool to see how the clones change over time, because over time the treatment can select for certain clones. And so they can see that then these patients underwent chemotherapy, intrathecal chemotherapy, and then they're following them early induction, mid induction, and then end of induction, and they're able to see this cell-free DNA go down, and then the patient clinically improve. But they're also able to identify how the clones are changing. And then the bottom panel, these are two patients who actually were found to not have, by flow cytometry, any cells in their spinal fluid. But interestingly enough, the cell-free DNA was, they were able to sequence and pick up some of these IGH clones, making a question about, is sequencing cell-free DNA going to be more sensitive than finding cells in CSF? This has a lot of implications for obviously ALL and relapse, but also for leptomeningeal spread. A lot of us know that radiographically we can be suspicious for leptomeningeal spread, and then we could do multiple lumbar punctures and not get any cells on flow cytometry or cytology. Are we going to change that paradigm that now we're looking for cell-free DNA? And is that what's going to diagnose leptomeningeal disease? I think that the answer is probably yes. In a couple of years, probably we are going to use different methods. So another thing we talked about is, what about the problem with, the blood is better to sample than CSF. I don't think I would want to undergo a bunch of lumbar punctures throughout my clinical care. And so this group did something really cool where they're using ultrasound to open up the blood-brain barrier for a period of time. You can see it in this bottom right. You can actually localize the ultrasound blood-brain barrier opening to an area that you want surrounding the tumor. And they were able to show that this increases the amount of cell-free DNA shed into the blood. And then you're able to do liquid biopsies from these brain tumor patients. And again, this is before opening up that blood-brain barrier, then after, you can see that increase in cell-free DNA, and they're able to follow and track these patients over time. And that may be another way to understand the treatment course. Again, this is very synergistic with long-read sequencing technology. I just want to touch upon, in closing, about sequencing in a developing world. this map is from 2016. We kind of joke about, oh, these brain tumor classifications require all the sequencing, and that's really relevant in America, Canada, and maybe, Western Europe. But in a lot of countries, you are going to get a brain tumor and they have no capability of sequencing and giving you these WHO 2021 guidelines. That is just not feasible in many countries. I think that's definitely going to change with long read sequencing for sure, because that is a portable device. And for example, Africa CDC used nanopore sequencing when COVID-19 came out to sequence their variants. And this was actually funded by the Bill and Melinda Gates Foundation, and Africa CDC worked together with Nanopore to set up these sequencing centers in Africa. So again, in developing countries, long read sequencing is definitely going to be a game changer. And then I think a lot of different countries are going to be emerging as giants in a genomics field. India is going to be the most populous country in the world. And this Anu Acharya, she is an amazing person who has companies in India on mapping genomes and using this to change healthcare and address disparities. So I think we're going to definitely not be so Western in our thinking. I think a lot of other countries are going to add so much to this growing field of using sequencing to change clinical paradigms. So what do I foresee the future paradigm being with long read sequencing integration? I think that we are going to get to a point that we're going to have intraoperative sequencing. There is a paper about using, looking at frozen tissue sections and using machine learning to predict the tumor diagnosis. But I think we're just going to go one step forward and just get the diagnosis from intraoperative sequencing, and then you know what DNA mutations are there and the molecular markers. Potentially you can do it while the person is operating and give feedback to the surgeon, and that will actually help with intraoperative decision making. I think you can actually get a methylome and transcriptome within 5 days if you use long read sequencing, and that will give you prognosis. I think the methylome is probably one of the best ways to give you prognosis over other types. So you can counsel your patient on what you think they're going to do, how they're going to do, and what treatment is best for them. And I think long term, I don't think MRI is going to go away, but it would be really cool if we can also just do liquid biopsies to sort out, is this recurrence? Is this radiation necrosis? Are you responding to your treatment? Let's just take a blood sample and let you know. So these future directions, I think real-time diagnosis is going to come with sequencing. I definitely think we have to standardize molecular marker testing and validate liquid biopsy results, because there's that question about, will the cell-free DNA show up before cancer cells, before we can identify cancer cells in the spinal fluid? Can we identify cell-free DNA earlier? And are we able to understand leptomeningeal spread earlier and treat these patients better, or relapse in the CNS for these ALL patients? So that, I think, will be the next step. We also need to address disparities. I don't really think it's fair that only certain countries you're able to get these really high levels of genomic sequencing done and help with prognosis and treatment paradigms. I think that precision medicine should be available to multiple people all over the world. And I think we're going to be able to address disparities. Long read sequencing is portable and doesn't require the same infrastructure as Illumina sequencing. And that kind of allows for you to sequence in various countries. And then again, we need to use this data to understand progression and resistance, and ultimately provide patients with personalized care. So thank you guys so much for your time, and I'll take any questions.
Thank you, Analiz. That was a tremendous talk and a great overview of sort of the modern state, as you said, of genomics for neurosurgeons when it comes to brain tumors. Although if that's for neurosurgeons, I might need to find a new Profession. So there's a question in the chat, which is, sample size and number of samples when you're talking about brain tumors and doing long read sequencing. So what sort of tissue mass or volume are you talking about? And if you're talking about small samples, what do you do in terms of potential sampling errors because of genetic heterogeneity within the tumors?
Yeah, those are good questions. So as I showed you from the cell-free DNA work, you can, I didn't actually tell, so there was one slide where I said that you can get, picograms. You can identify mutations with picograms of DNA. Interestingly, you can go down to femtograms as well. So I think that just one biopsy core is enough to sequence off of. Yes, I do think heterogeneity is still an issue, because you could say, for example, send a core that's radiation necrosis. Oftentimes radiation necrosis is mixed with tumor, and miss a core that's not. But I would say that that kind of holds true for current histopathology as well, in terms of, are you just looking at the certain region of the tumor? And then what's also, I forgot to tell people, is with current Illumina sequencing. So if you have a slide, an H&E slide, and you send it off for Illumina next generation sequencing, in order to get the information back in regards to DNA mutations, ideally you want to have at least 70% of that sample be comprised of tumor. So you can't just send, say it has 30% tumor and 70% radiation necrosis. You won't be able to sequence with Illumina. You won't be able to get information back. So I think the way to address the heterogeneity is perhaps we use a lot of like 5-ALA and things like that. I really think that will come more into play using maybe different fluorescent dyes to identify regions of tumor versus treatment change. And then within tumors, there's still heterogeneity, and perhaps we can come up with paradigms about, should you sample different regions, and should that be a standard that you're sampling different regions of the tumor to address that heterogeneity.
Thanks. I want to pivot to what you touched on towards the end of your talk in terms of healthcare disparities. And aside from the disparities internationally, we have disparities within our own communities. And I'm curious your thoughts about how we lower the barriers to access to this level of care for people in our own country, in our own communities, and some of your thoughts of what you've even tried to do in Arkansas to, try to get the most number of people the access to this level of medicine.
Yes, so I work in obviously a rural, poor state, and addressing healthcare disparities is something that I want to do personally. So one thing that we do is that we do offer next generation sequencing to all of our patients. However, we use a company, and I'm not, as you can see, my disclosures, I do not work for this company, but so there are various companies available for next generation sequencing for tumors. Caris, Foundation One, Tempus are the most common three. They each have their pros and cons. I don't want to go into that either. But we have used predominantly Tempus for a couple of reasons. One, they have a very generous financial aid program. And so that's why we use them, because a lot of our patients, their insurance would not cover this. And then two, they also have a mobile lab where if you want a blood sample, they will actually send someone to the person's house and pick up a blood sample. They also, the financial aid program covers the patient for up to a year and sometimes longer. And so if, for example, it's a lung cancer patient and they need multiple liquid biopsies, they would still qualify to get those done without paying. So that's one way we've addressed it, because our patients a lot of them are underinsured or uninsured.
Thank you. Any other comments or questions from our panelists?
Charles has his hand up there, Zach.
Oh, sorry, Charles.
Yeah, this was a great talk. I really thought it was wonderful. And I think that you have wonderful slides and are looking into the future of the way we'll be taking care of these tumors. We actually here at Swedish were trying to purchase a nanopore machine, and that didn't work out. But my colleagues, now that they know about long read sequencing, I just want to let them know we actually have a NCI grant where we are actually doing long read sequencing currently of normal tissue surrounding GBM and GBM, and looking at alternative splicing from the RNA. And it really was helpful for you to explain how all this works, because what we're finding are that there's novel splice variants that encode for novel epitopes that could be neoantigens for immunotherapy. So the whole field is being accelerated. We send that off to PacBio. We were trying to get the nanopore machine, but it didn't work out. But your explanation and description is very timely. I think that the field will be turning. So that was a great talk. Thank you.
Yeah, you're welcome. And I will say a little caveat about the nanopore, even though it was like, oh, it's so simple and you just plug it into your computer. The biomedical informatics that's needed to deconvolute those signals is not easy. I do not do that. You definitely need to have really, it's getting better, but it's not like just, you know, your regular, like us, are the clinicians are just going to be like, you know what, I'm sequencing my patient myself, and you're plugging in the USB and you're deconvoluting signals and getting readouts and mapping back to, you know, a reference genome on your own. That, I don't think it's to that level. So you do need some biomedical informatics support, and I didn't touch upon that.
Yeah, we have a group in Oregon, Portland, that's part of Providence that does really good genomic bioinformatics. So they've been very helpful. Thank you.
Analiz, there's a question in the chat about potential therapeutic targets as a result of this level of genomic understanding of tumors. And then I would ask you to comment relatedly, how do we build either registries or data sharing agreements so that we don't have fragmented genomics of these tumors, but we get big data to guide some of the therapy moving forward?
Sure. So I think that this technology is really powerful because you can just do everything in together. For example, like you can do DNA and RNA and methylation pretty much very easily with using the same platform. In regards to how it may change drugs, so the other thing I touched upon very briefly was that now these nanopores you can also do protein. So then you can think that you're doing multi-omics. You can do genomics, transcriptomics, epigenomics, epitranscriptomics, and proteomics using the same platform. And then you can also use this, I didn't go into this much, for single cell sequencing. And so I think that overall it's just much more powerful. But I guess in combination with, I think our preclinical models, so I think, you know, ex vivo organoids and xenografts, and then trying to build these high throughput systems where you're screening you know, drugs on these preclinical models, and then you're sequencing and getting these readout and building these like predictive paradigms of what is a responder and what's a non-responder. I think the biggest caveat is all of that needs clinical patient level validation. And I think that's why it's important for there to be this translation where this technology is explained to the clinicians, and we can say, oh my God, that would be so powerful. like that intraoperative diagnosis, I bet like a lot of neurosurgeons around here would be like, that would change what I do in the operating room. Yeah, I think it's just such a powerful technology, but yeah, the clinical validation and building the preclinical models for the drug testing, I think maybe one of the main barriers, but this technology would be able to do everything that you would need, all the omics, as well as even on the single cell level. And then I think you asked me, I forgot what your question was, Dr. Litvak. You asked,
Yeah, so the therapeutic targets.
Okay. Yeah, I guess I think you were talking about, so how we would work together. I think I agree with you that we probably would need to build consortiums and have things that are very clinically annotated, meaning these large consortiums, similar to how I showed you that a group in Germany that has this like methylation consortium, Dr. Zade's group with her work with the meningiomas, they built a consortium in order to get all these samples. So I think that's definitely what we'll need, and then have this high level of clinical annotation so we can understand, okay, how did this patient respond? And was our prediction correct? And I think that's how we can get that clinical validation to understand, are we really identifying targets that are clinically useful? Because again, just doing it for research purposes, that's really cool, but does it translate to improving a patient's outcome? I think that's where we want to go.
Awesome. Well, thank you for being so generous with your time this morning. Appreciate everybody connecting. For those of you who are attending for CME purposes, we'll have the slide up for you to log CME, and wish everybody a happy Thursday.
Thanks so much. That was a spectacular presentation. Really appreciate it.
Thank you.